Hi students, hope you're all doing well. Today we're going to study the key factors that control the speed of enzyme reactions. You know, these tiny engines that power our cells are amazing, but what happens when you start messing with their environment? We're about to find out what can supercharge them or shut them down completely.
So here's the plan. First, we'll map out what a typical reaction looks like from start to finish. Then we'll get into the two main ingredients, the enzymes themselves and their fuel, the substrate. After that, we're going to turn up the heat, literally, and see how temperature and pH totally change the game. And finally, we'll pull it all together and see how it's all one big balancing act.
Okay, so before we start fiddling with all the variables, let's just establish a baseline. What does a standard run-of-the-mill enzyme reaction actually look like? What's its signature pattern?
Check this out. Pretty much any enzyme reaction you see is going to follow this classic curve. It just explodes out of the gate with this incredible burst of speed. I mean, look at how steep that line is at the beginning. But then you can see it gradually starts to slow down, and eventually it just flattens out completely. So why does that happen? Well, it's literally running out of fuel as the substrate gets used up.
Now that very first moment, that initial super fast burst, that's what scientists really care about. It's called the initial rate of reaction. Think of it as the purest measure of the enzyme's true power, before anything else has a chance to slow it down. It's the best way to make a fair comparison of its performance under different conditions.
Here are the core details regarding this topic. Review them intently for their team crucial for your finals.
So let's ask the big question. Why the slowdown? I mean, it's not like the enzymes are getting tired, right? The answer is all about the ingredients in the mix, which brings us perfectly to our next two factors. All right, let's look at our two key players here. The enzyme, that's our worker, and the substrate, which is the raw material. How does having more or less of each one really affect how fast this whole process goes?
So first up, let's talk about the enzyme concentration. A great way to think about this is like checkout lanes at a grocery store. The active sites are the lanes, and the customers are our substrate. If you've got tons of customers waiting, what happens when you open more lanes? The whole process gets faster, right? It's a really simple, direct relationship.
And when you actually plot this on a graph, you get a perfectly straight line. The relationship is what we call directly proportional. If you double the number of enzymes, you double the reaction rate. Simple as that. Of course, that's assuming you have plenty of substrate to keep them all busy.
Okay, but what if we flip that around? What if we keep the number of enzymes the same, but we just keep adding more and more substrate? At first, yeah, the rate's going to climb, but it doesn't just keep going up and up forever.
You can see it clear as day on this curve. The rate goes up, and then, boom, it hits a ceiling. It just plateaus. This is its maximum speed. At this point, every single enzyme is working as hard as it possibly can. Adding more substrate now is like trying to cram more customers into a store when every single checkout lane is already occupied. It won't make things any faster.
I absolutely love this quote from the source material because it gives you the perfect mental picture. The substrate molecules are literally forming a line, waiting for an active site to open up. The enzymes are completely saturated.
This maximum speed, this ceiling, actually has a specific name. It's called Vmax. It represents the absolute top speed that enzyme can hit under those conditions. It's a fundamental limit, kind of like the red line on a car's engine.
Okay, let's switch gears from concentrations to something we can all feel, temperature. You know, heat can be an enzyme's best friend, or it can be its absolute worst enemy.
This graph shows a really critical relationship. You can see the reaction rate climbs as it gets warmer, but only up to a point. Once it hits that peak, the rate doesn't just level off, it drops off a cliff. It absolutely plummets. Why? Because the heat is actually starting to break the enzyme itself apart.
That peak, that sweet spot, is called the optimum temperature. For us humans, our enzyme's optimum is right around 40 degrees Celsius. It's no accident that our bodies work so hard to maintain a temperature of 37 degrees. It's to keep our enzymes humming along happily. It's also why a really high fever can be so dangerous. It pushes our enzymes past that breaking point.
So let's break down what's happening on either side of this curve. On the left side, down in the cold, all the molecules are just sluggish. They're moving too slowly, so they don't collide very often. But on the right side, where it's too hot, it's a completely different problem. The heat makes the enzyme vibrate so violently that it literally shakes itself apart. The bonds break, it loses its special 3D shape, and it just stops working. We call this denaturation, and it's permanent. There's no coming back from that. Now, for our last major factor, and you'll see it behaves in a way that's surprisingly similar to temperature. I'm talking about pH. Turns out, an enzyme is just as picky about the acidity or alkalinity of its environment.
And would you look at that? It's another bell-shaped curve. Just like with temperature, there's a perfect optimal pH where the enzyme does its best work. For a lot of enzymes, that spot is right around a neutral pH of 7. But you have to remember, every enzyme is a specialist, built for its own unique environment.
And that peak performance point is, you probably guessed it, the optimum pH. Now, while many enzymes love neutral conditions, some are built for the extremes. For instance, the enzyme pepsin works inside your stomach, which is incredibly acidic, so its optimum pH is way down around 2.
So why does pH have such a dramatic effect? It all comes down to electrical charges. Extreme pH levels, either too acidic or too alkaline, mess with the positive and negative charges on the enzyme. This disrupts the delicate ionic bonds that hold its perfect three-dimensional shape together. And just like with too much heat, if you stray too far from that optimum, the enzyme denatures.
Listed are the main insights for the heading discussed. Note them specifically since they are highly relevant for exams.
So, as we've seen, it's not just about one thing. For an enzyme to work properly, it's this incredibly delicate balance of all these factors working together in harmony. Let's do a quick recap of the key points. This little table sums it all up beautifully, doesn't it? More enzymes, you get a direct, straight-line increase in the rate. More substrate, the rate goes up until it hits that ceiling, Vmax. And for both temperature and pH, you've got that sharp peak, that optimum point, where performance drops off a cliff on either side because the enzyme's actual shape is on the line.
And this all leads us to a final, much bigger thought. We've just spent all this time looking at each of these factors one by one, in isolation. But think about it. Inside a living cell, all of these things are happening at the same time. How on earth does a cell manage to control the perfect temperature, the perfect pH, and the right concentrations of everything to keep thousands of different enzymes working together? Well, that is the incredible magic of homeostasis.
And that's our look at the factors that control enzyme action. Hopefully this gives you a much better feel for just how dynamic and, honestly, sensitive these little cellular machines really are. Thanks so much for watching.
Key vocabulary related to this topic is displayed, sorted according to multiple exam boards. These words require serious attention during preparation because of their importance and must never be neglected. In addition, analysis of past paper is provided for each exam board and paper style. Focus on this carefully as it will greatly assist in improving your preparation. Try to understand the context and practical use of each keyword rather than learning them by heart alone. Frequent practice with these terms in past questions will enhance your accuracy and overall performance.
Please subscribe our channel to get more educational videos. Thank you.